Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. Now the purpose of these videos is to provide a very rudimentary and fundamental understanding of some basic concepts in microbiology for what would be considered sort of a freshman to sophomore level microbiology course. Now there's a tremendous amount of detail that we could go into and at times maybe should go into, but as you know I've found too much information too soon can lead to information overload and stress people out. So know that whatever class you're taking with whatever instructor you're taking, they may require additional information or provide this in a different format or different presentation order, but ultimately the information that I'm going to cover is sort of the foundational principles in order to understand the more difficult concepts. So I'm going to simplify a few things. Now today's lecture is going to be the beginning of what we call metabolism. So I'm going to go through a lot of definitions in this lecture and try to explain what some of these words mean. This will be a series of 2 to 3 lectures, and the additional lectures will actually go into some of the chemistry of what's going on from a rudimentary standpoint. Okay, so let's get started. Now when we talk about metabolism, technically what we're talking about in metabolism is we're talking about the sum total of all biochemical reactions in an organism. So if we were talking about human metabolism, we'd talk about all the chemical reactions in our bodies. If we talk about microbial metabolism, we're talking about the metabolism in little microbes, bacterial metabolism, and so on and so forth. Now, usually, what we're going to talk about in these biology classes is energy metabolism. And this is basically all the biochemical reactions in the production of high energy compounds. And the number one high energy compound we're going to talk about is ATP, which if you're not familiar with stands for adenosine, that's the A, triphosphate, that's the T and the P. And essentially adenosine is a ringed molecule that has some nitrogen in it. It's very similar to adenine from adenine, thymine, guanine, and cytosine. I'm not going to draw the structure out, but it's a ringed molecule. And if I stick a single phosphate group on it, when I talk about a phosphate group, I'm talking about what's called a PO4 with a negative charge, okay? So this would be attached to some carbon-based molecule called adenosine. Now, if I stick a single phosphate group on there, I have what's called AMP, adenosine monophosphate, mono meaning one. If I have adenosine with 2 phosphates stuck somewhere else or stuck together on it, then I have what's called ADP, or adenosine diphosphate, di meaning 2. If I have adenosine with 3 phosphate groups attached, then I have what's called ATP, adenosine triphosphate. And essentially the significance of this is this. Every one of these covalent bonds between the adenosine and the phosphate, or the first phosphate and the second and third phosphate, it requires energy to form those chemical bonds. I have to make these things come together by putting some energy into them. Kind of like in a human relationship. Sometimes you got to put energy into forming a bond with someone. Sometimes it happens spontaneously. But when we put energy into that bond, when we break that bond, the energy can be released. And we're talking about, energy in the sense that it's really hard to define energy. I dare you to ask someone to define what energy is. And we can't, it's an abstract concept. We can't see and touch and feel energy, but we can describe it mathematically in physics and things. But nonetheless, it's the ability to do work. And so when we're talking about energy, if I need to move something around or form another molecule, it may take energy to do that. Our cells will use this last little bond between these last 2 phosphates for energy. And the reason is this bond releases more energy than it took to form the bond This bond releases a little bit more energy than it took to form the bond, and this one releases some energy when we break it. But the one that gives us the greatest amount of energy is this third little bond between these last two phosphates. Considering how much energy it takes to put in, when I break it, it releases the most. The best example I can give you, instead of talking about kilojoules and joules and types of energy that you talk about in chemistry, is what if I used monetary amounts, dollar amounts? Let's say it takes 98 cents to form this energy, form this bond, I should say, but when I break it, it gives me $1 worth of energy. This isn't exactly how it works, but it's a simplified concept. And that would be the same for the first phosphate bond in any of these. Now, let's say this one takes, let's say, 88 cents. And when I break it, it gives me $1 worth of energy. Again, this is just a simplified example. And let's say this last bond takes about 8 cents, but when I break it, it gives me $1 worth of energy. Which bond releases the most energy? This one. It gives us the biggest bang for our buck. We would get 92 cents for every 8 cents we put in, where here we would only get 12 cents, and here we only get 2 cents. It's almost as if you went to a bank. If I said for every 98 cents you invest, I'll give you $1 back tomorrow, you would make money. For every 88 cents, I'll give you $1. Which bank would you pick to invest in? I'd invest in this one. If I told you for every 8 cents you give me, I give you $1 back tomorrow, then most people would be wise and invest in this bank. If I run out of this, I could use this and this, but they're almost not worth the trouble, so to speak. Now, that's not exactly how it works, but it's a pretty good example to wrap your brain around. And I need my eraser, if I can find it, so I can erase some of this, okay? So that's why adenosine triphosphate is a high energy compound. It releases the highest amount of energy when we break that little phosphate group, okay? Now, when we talk about metabolism, we really usually are talking about energy metabolism, which is what are the chemical reactions that are involved in the production of ATP, or energy for the cell, okay? And I misspelled compounds there, but that's okay. So, now, when it comes to metabolism, there's different types of chemical reactions we can talk about. One of the chemical reactions we can talk about are called catabolic reactions. And I'm going to abbreviate the word reactions with an RxNS. So when we talk about catabolic reactions, or catabolism, this comes from a Japanese word, kata. Like katana means the sword. It means to cut. So catabolic reactions are sometimes referred as decomposition reactions. To decompose means to break down, so I think of these as breakdown reactions. This is the way that I simplify it for my brain. So I can take a large molecule, like substance A, and if I run it through a catabolic reaction, I might get several smaller molecules out of it, okay? If I break one thing into multiple things, I have what's called a catabolic reaction, or decomposition reaction. Some people call them digestion reactions, okay? Now, the opposite of this would be what we would call an anabolic reaction. Ana means up or with, to build up. So anabolic reactions are what we sometimes refer to as synthesis reactions in chemistry, or I call them build-up reactions. So essentially, if I took several small compounds, compound M and Q plus R, and I run them together and make some really large molecule, let's say I make substance Z out of 3 smaller ones, then that would be an anabolic reaction. Essentially, a catabolic reaction is when we break a large molecule into smaller ones. And an anabolic reaction is when we build a large molecule out of smaller ones. So we can think of this as we break large molecules into smaller ones. And here we break, oops, no we don't, we make larger molecules out of smaller ones. So here we're breaking down, and here we're making larger, we're putting things together. So those are some basic types of reactions that we're going to talk about. Now, a couple of other things we need to talk about is, for one, what are the reasons that we're going to undergo metabolism? Well, there's two reasons. Number one is that in some of these reactions, we're going to release energy. So energy production is a major one. Another thing we could do is we can replenish what's called the nutrient pool. The nutrients are those compounds we're going to break down for energy. So now, before I go into some of the notes that I have produced for my students anyway, I'm going to do a basic overall review of the four major organic compounds and how they can be used for energy production. So we have four major organic compounds that we talk about in biology. And we've done this in a previous video, but one is called carbohydrates. In plants, it's called starch. In animals, it's called glycogen. Now, another name for carbohydrates is polysaccharides. Poly means many, and saccharide means sugar, like saccharin. So polysaccharides means many sugars. So essentially, I can take a whole bunch of sugars and I can put them into chemical bonds and form some really large molecules out of these things and bind these sugars up. And the more I have them bound up together, the harder it is to burn them. Okay? So imagine a three-dimensional block here. Now ultimately what I want to do is I want to use enzymes called Saccharadases. And here's something that I want to talk about, and we're going to do this a little bit later in the video. We're going to talk about enzymes. What are enzymes? Enzymes are usually proteins that increase the rate of biochemical reactions. Or another way to say that is we can say that they lower the energy of activation for a chemical reaction. Okay? So before I finish this, let me just show you a quick graph. Okay? If I do the amount of energy it takes for a chemical reaction to occur over time, I'm going to start off with what I call reactants. Okay? And so let's say I have substance A plus substance B, and I'm going to track this chemical reaction. And since the energy is going to be increasing from 0 all the way up, then it might take a certain amount of energy for this reaction to occur over time to come together to form substance C. And we would call this energy right here the energy of activation, or the activation energy. How much energy did I have to put in to make this chemical reaction occur? Now, what if I did this with an enzyme? And NZ is an accepted abbreviation for enzyme, or at least it's mine for my notes. So in the presence of an enzyme, so now I add the enzyme in, and look at the energy of activation. It takes less energy to make this happen. So I don't have to wait all this time for it to occur. I can shorten the reaction time. And I didn't shorten the time on my graph, but nonetheless, that's all that enzymes are. Enzymes are proteins that can make chemical reactions happen more easily and more rapidly at the speed of life, so to speak. For example, If I took a whole bunch of amino acids and I wanted to build a protein out of them, or if I just wanted to break down sugars, it might take several hundred years for these sugars, or several hours or days for these sugars to break down. But what if I need them instantaneously? I need them within a matter of seconds. Well, if I add an enzyme in here, then I might break this large sugar down into individual sugars called monosaccharides or simple sugars. Okay? So I'm going to erase all of this. We don't really need this right now, but that's what enzymes do, is they lower the energy of activation, or the activation energy, or they speed up the rate at which a chemical reaction or a biochemical reaction can occur. And so the enzyme that's involved in this reaction would be called a saccharidase. Saccharadases are a whole class of enzymes that break polysaccharides into monosaccharides. Now, one of the most well-known monosaccharides we can talk about is glucose. And if you look at the chemical formula of glucose, it's C6H12O6. Now, the reason this becomes important is if I combine this in the presence of oxygen and some enzymes, what I get out of this is going to be 36 molecules of ATP. That's a whole lot of energy. I'm also going to get some carbon dioxide and some water. Now, I haven't balanced the equation. Let me make sure I'm not writing off of the screen. Yes, I am a little bit, so I'm going to tilt my camera, okay? But if you look at this chemical reaction, and we're going to write this over again in a little bit, but ultimately I need sugar to make ATP. So let me rewrite this a little bit nicer and bigger. One of the monosaccharides that we can discuss is C6H12O6, which is called glucose. Ose usually means sugar at the end of a word. Glucose, fructose, lactose, mannose, hexose. Anything that ends in ose is usually a sugar. Anything that ends in ase is usually an enzyme. So there's sort of a shorthand or a language to chemistry and biochemistry. Anyway, if I take this in the presence of oxygen, and really if we wanted to balance the equation, I would use 6 oxygens in the presence of some enzymes. And those enzymes are usually inside of an organelle, inside of eukaryotic cells called the mitochondrion. In the presence of the mitochondrial enzymes, I can convert glucose and 6 oxygens into 6 carbon dioxides, 6 waters, plus 36 molecules of ATP. That's the big kicker. If I take the oxygen out of the equation, I only get 2 ATP, and we're going to talk about that in some more detail. But ultimately, one of the 4 major classes of organic compounds, called carbohydrates or polysaccharides, is how we store sugars in nature. In plants, they store it as starch. In animals, it's glycogen. When we eat plants or animals, we break that down using saccharidases into monosaccharides, simple sugars like glucose, and then we burn that for energy. And if I get more sugar than I need, and I'm a plant or an animal, I can convert this back into glycogen or starch, depending on the organism, using enzymes. Now I'm going to erase this definition of enzymes for you because we already have it down, and we're going to do this some more in a little bit. Now there's a second major class of organic compounds that we can talk about. I'm going to use a different color. There are lipids. Lipids are known as fats, oils, or waxes in nature. And one of the things that happens when we break down lipids through a series of chemical reactions with enzymes called lipases is I can get 2 major substances out of this. I can get these things called fatty acids, and I can break them down into a glycerol. And if you look, anytime we have gluc or glyc, it has something to do with sugar. The all means it's an alcohol form. Now, really what I want to use lipids for and these structures for is sometimes for biological membranes. So when I eat fats, I can rebuild cell membranes and heal and grow cells and grow the membranes and repair them. But if I'm out of sugar and I don't have anything for energy, these guys can be converted into glucose or a monosaccharide and used for energy. So the primary source of energy in our cells is polysaccharides and simple sugars like glucose. Lipids can be broken down with molecules called lipases, enzymes, and they can be broken down into fatty acids and glycerol, particularly a class called triglycerides. Other lipids just are used for membranes and for storage of fat. But one of the reasons we store fat is that not only can we heal and grow biological membranes, but it is an alternate source of energy. If I run out of sugar, I can burn fat, which is, by the way, the secret behind all the Atkins diet and Mediterranean diet and paleo diet and all of the low carbohydrate diets. You cut out carbs, your body burns the fat until you get down to the weight you like, and then you reintroduce healthy amount of carbs. Eating carbs, since I can convert carbs or fats into sugar, I can convert sugar into fat in the reverse process. And what's making America and the world fat and obese is not the consumption of fats, but the consumption of sugar. So we drink way too much sugar in our sodas and everything else. But anyway, now there's a third major class of organic compounds. So I'm going to erase this because I got to keep writing my organic compounds down, and I don't have a large enough board or enough space. But ultimately, you guys know that we use polysaccharides to break down the sugar. We can use sugar for energy. I use lipids for biological membranes for growth and repair, or and cell division, or I can use them as an alternate source of energy. Now, a third major class of organic compounds we can talk about is called proteins. Proteins are also referred to as polypeptides. Now, proteins are considered to be long chains of molecules called amino acids. And amino acids, I will abbreviate as an AA. If I stick them together in a long chain like this, and I take a couple of little A's and put a circle around them, each amino acid is called a peptide, because the bond between them is referred to as a peptide bond, and we'll talk about that later. So when I eat protein from animal muscle and things, this is what I'm getting, is these long chains of amino acids, usually somewhere between 50 and several hundred amino acids long, depending on the protein. By the way, all proteins in nature are made out of the same 20 amino acids, whether it's a bacterium, whether it's a human, whether it's a roach or a goat or whatever. We use the exact same 20 amino acids, plants, bacteria, everything. The only difference that makes one protein different from the other is the number of the amino acids and the arrangement of the amino acids. Now, when I eat amino acids, I'm going to digest them in a series of chemical reactions using enzymes called proteases and peptidases. And these are classes of enzymes. There's a bunch of individual ones. But notice there's the ASE in the name, and usually part of the name tells you what the enzyme works on. Now, when I digest proteases and peptidases, what I get, or proteins, using proteases and peptidases, is I break down these peptide bonds and free up the individual amino acids. So I can convert chicken muscle into human muscle. If I'm trying to build mass, I want to eat a lot of protein. And so we can replace all the proteins in our body, or any organism can replace proteins that are broken down over time by consuming proteins from an outside source and then breaking them down into amino acids. Now, there's two uses for amino acids. One is protein synthesis. The ribosomes of my cells are going to take amino acids and rearrange them into the proteins that my cell needs. All proteins are made out of these amino acids. I just need the messenger RNA that tells me what order the amino acids go in. But the second use is if I remove the amino group, which is called deamination. then I can use the carbons, hydrogens, and oxygens in the amino acid to convert it to sugar for energy. So now I can use lipids or amino acids and proteins as an alternate source of energy. Okay? Now, one of the things I didn't cover with you is this. We know that carbohydrates are made out of carbon, hydrogen, and oxygen. Lipids are made out of carbon, hydrogen, and oxygen. Those are the only atoms in most lipids. Proteins are made out of carbon, hydrogen, oxygen, and nitrogen. I'm going to put sulfur in parentheses because there's 2 amino acids that have sulfur in them, so they usually have some sulfur in the protein. But proteins have carbon, hydrogen, oxygen, and nitrogen. And it's that nitrogen group, the amino group, that we have to get rid of. And that's what gives us the amino in amino acid. Now, I'm running out of space, so I'm going to erase some of this, okay? But one of the things I want you to see is that these 2 organic compounds can be converted into sugar for energy. That's part of metabolism. If I run out of sugars and I don't get a chance to eat today, if I starve myself, then my body will start to burn fat. When I run out of fat, my body will start to burn proteins and amino acids. In the process, I have to remove the amino group or the nitrogen-containing compound, which is going to give us the nitrogen waste, which can be bad. So, if you can keep this in mind, since I have a small board, let me do one last thing. Well, let's see if I can squeeze it right here. The 4th major organic compound is called nucleic acids. This would be DNA and RNA are the nucleic acids of our cells. And let me make sure this is all within the view of the screen. Yes, perfect. So, nucleic acids are made out of carbon, hydrogen, oxygen, and nitrogen. If I digest nucleic acids, I'm going to get 3 things out of a nucleic acid. I'm going to get a 5-carbon sugar. Let me erase that. That didn't come out right. I'm going to get out of this what's called a 5-carbon sugar. That 5-carbon sugar is either going to be deoxyribose or ribose. And look, if I do some chemical manipulation with some leftover carbons from somewhere else, I can convert that 5-carbon sugar into glucose if I wanted to. I'm going to get a phosphate group. Well, this phosphate is the P in ATP. And the last thing I'm going to get out of this, and since I'm running out of room, let me make sure I can see this on the screen. Give me a second. It's just me with my camera. Yeah, we got plenty of room over here. So the last thing I'm going to get out of this from these 3 compounds is a nitrogenous base. A base simply means it has a pH greater than 7 and it has nitrogen in it. This has carbon, hydrogen, oxygen, and nitrogen. So technically I could take a couple of the bases and I deaminate. I remove that amino group. And I have carbon, hydrogen, and oxygen. With some chemical manipulation and some enzymes, I can convert some of these compounds into sugar for energy. And this is how I think about it. I think of sugars as cash money. And if I have a whole bunch of cash, I don't want to walk around with $1,000 in my pocket. So I would take the amount that I might need. Let's say I take $100 out. The other $900 I stick in my bank, and that sugar bank would either be starch if you're a plant, or glycogen if you're an animal. And then I burn my cash. When I run out of sugar, I can tap into my sugar bank and release more glucose. I can break down the carbohydrates stored in my body, in muscle and liver, or in plants, and the energy reserves in the plant, in the starch. But what happens when if I don't continue to eat and get energy, then what happens when I run out of sugar? Well, when you're out of cash and you have no savings account left, you start pawning things and selling things. The first thing I could get rid of is the stuff that I want the least, which would be fat. So my body can convert fat into energy if I need it. But it's not always as efficient, or it's not always the preferred process, although it can generate quite a bit of energy. When I run out of fat, so if you go to your house and say, I'm out of money and in order to eat tomorrow I have to sell some things, you look at the things that you utilize the least. Maybe a VCR or a DVD player or a stereo or things, and you start getting rid of things that you never use anymore. An old bicycle, take it to the pawn shop. When you run out of that stuff, if you still haven't gotten a source of energy, you haven't eaten, your body will start to burn proteins. I think of getting rid of protein as of getting rid of things that are functional in your house, like selling your car, selling your microwave, selling your stove, selling your refrigerator. Now you don't have anything that can function, and it's really difficult to survive. We don't want to do this unless we absolutely have to. And then the last thing we do is we start selling nucleic acids. That's when you're selling blood and things from your body, and you can't survive very long. This is when we get into starvation. We go into breaking down fat and then protein, and then once you're here, you're pretty much going to die. So those are the basics of the 4 major organic compounds. And I would prefer to use proteins for protein synthesis. I would prefer to use lipids for biological membranes, growth and repair and stuff, and reproduction, cellular reproduction, cell division. I would prefer to use nucleic acids as the DNA and RNA in my cells for repair and damage or cell division and things to make new cells. But if all else fails, I could use each one as an alternate source for energy, because ultimately if I don't have energy to do things, none of this other stuff matters. Now that's sort of, oh, and by the way, the enzymes that do this stuff are called nucleases. Again, you see ASE at the end of the word means it's an enzyme. I have proteases and peptidases, I have lipases, and I have nucleases and saccharadases that digest the 4 major organic compounds to ultimately give me these other molecules for use. Sugars are used for energy. Lipids are used for membrane growth and repair and cell division, but they can be used for energy if I'm out of sugar. Proteins, the amino acids I want to use to make new proteins to replace the damaged proteins in my cells and grow my cells. But if I have to, I can also, by deaminating or removing the amino group, I can convert them into glucose for energy. And then when I break down nucleic acids using nucleases, I can get a 5-carbon sugar. Now I got to spend energy manipulating it and adding more carbons and hydrogens to get to a 6-carbon sugar, but I could do that. The phosphate can be put into the phosphate of ATP, and then the nitrogenous base has nitrogen in it. I have to deaminate or remove the amino group and convert it into glucose or a simple sugar, so then I can get energy. And by the time I'm here, I'm dying. I'm not going to survive, okay? Now this is what happens in a lot of eukaryotic cells and even in some prokaryotic cells, and we're going to talk about this. Now, before I go any further, a couple of things that I want to talk about before I get into the rest of the notes that are listed for my students. I'm going to erase all of this stuff. Before I go much further, I have to talk about the deamination and the amino groups, okay? Excuse me. As I lecture, my voice gets messed up and my throat gets dry, and I either have to swallow or drink something. So, now, all amino acids have this structure. They have a centrally located carbon atom. And that carbon atom can have up to 4 covalent bonds. 2 of these groups we're going to call the R group and the R prime group. And it doesn't matter which one's R or R prime. And these kind of can be a number of other things. Other carbons and hydrogens and oxygens and other things. On one side, I'm going to have another carbon with this grouping. A C double bond O and an OH here is called a carboxyl group, or it's also called carboxylic acid, because it'll give off that hydrogen very easily, and once it gives off that proton, it'll become this, okay, a negatively charged acid, okay. So these two are in a bond together, but I can release that proton and keep the electron here, so it's almost like a ionic bond, so to speak. Now on this side, I'm going to have a nitrogen with 2 hydrogens. And this H2N with the negative charge is called an amino group. So if you look at the name amino acid, I have an amino group on one side and an acid group or carboxylic acid on the other. And then these parts can vary. And that's what makes each amino acid different from all the others. If I break this chemical bond here, I will release the amino group. I can combine that with another hydrogen atom, or ion I should say, a proton, and form NH3, which we call ammonia. And so sometimes when we're breaking down a whole lot of amino acids, there's an ammonia smell to urine and things. And that's when you've gone into some you can get into some bad chemistry. You don't ever want to smell that. And that's one of the dangers, by the way, of some of these low carbohydrate diets. The keto diet is because we can start to develop what are called ketone groups, and you go into ketosis. That means you're burning your proteins also. You're getting dangerously low on fuel, and it can be dangerous. That should be monitored by a physician. And ultimately, if you start to smell ammonia when you urinate, you're going to have some kidney problems. Anyway, we don't want that because ammonia is poison. These can be toxins that we need to urinate out, and that's what helps form urea. So I wanted to talk about the amino acid because a little bit later we're going to talk about the deamination also, and the formation of ammonia and other nitrogen compounds. It's going to be important in micro. All right, so now we've gone over the 4 major organic compounds and what they are used for. I hope that that made some sense to you. Now, if we go through the notes as they're listed, we can continue down the first page in this lecture. So it asks us, what is the basic overall equation of energy production or metabolism, energy metabolism, and we did that. C6H12O6 plus 6 oxygens and so on and so forth. Okay, we're going to come back to that a little bit later. So one of the questions that asks is, what are enzymes? Enzymes are usually, there's a rare occasion that this doesn't prove true, but they're usually proteins involved in lowering the activation energy of biochemical reactions. Or we can say that they are proteins involved in increasing the rate of a chemical reaction. So sometimes they make chemical reactions happen much more quickly. One of the things that's key about these is that they sometimes are called biological catalysts. A catalyst is any substance that can lower the activation energy of a chemical reaction or make it happen faster. But in biochemistry, we talk about enzymes, biological catalysts. Okay? And always in a catalytic reaction or an enzyme reaction, the catalyst or the enzyme is not used up in the reaction. So for example, if I have substance A plus substance B, and I can use an enzyme to make it happen at a much lower energy at a faster rate to get compound C, when I'm done, the enzyme can float off and catalyze another reaction. It's not used up. It doesn't get altered in the reaction, okay? Usually. So, now, a couple of more questions, okay? When we talk about enzymes, we know that these are long chains of amino acids because they are proteins. So when I talk about the primary structure, and primary is abbreviated as a 1 with a little odd. When we talk about the primary structure of a protein, we're talking about the straight amino acid sequence. What is the order of the amino acids? So if I have a protein that looks like this, and I know that this is amino acid number 1, 2, 3, 4, and so on. I'm not going to put the numbers down, but if I have a whole bunch of amino acids strung together in a long chain, and I know the order of amino acids, then I know what's called the primary structure of the protein. That's not the final structure. And the reason is, as we talked about before, each amino acid can have those R groups on it, and sometimes the little R group of one amino acid might have a high affinity or a desire to bind with the R group of another amino acid. And because they're floating around in this liquid environment, I could actually bring those two amino acids together and form a chemical bond here. And that might bring a couple of other amino acids close together that might not have been close before, and now this protein can fold up like this. And it'll start a series of chemical reactions, almost like those little foldy things, those round ones that you use as a shade for cars. I live in South Texas, so you shade your car, or otherwise you sit down and you burn your legs because of the heat cooking your seats all day. But once you start to twist those things, they just collapse down on themselves. Or sort of like pushing the button on an umbrella. They spring up. Well, proteins will automatically start to fold up until they get to this three-dimensional structure with all these chemical bonds inside. And it's this 3D structure that causes them to take on their activity or catalytic activity. This would be the functional form, or what we call the natured form. We go from the primary structure to what's called the tertiary structure. The tertiary, or third degree of folding, is called the natured form. This is how the protein exists in nature and how it does its job. So when I talk about the primary structure, I'm talking about the amino acid sequence. When I talk about the tertiary structure, I'm talking about a folded protein, the natured form. So we say that it is folded or natured, okay? So I'm going to erase some of this, and we're going to talk about that. But at least that picture gives us the idea of what we mean when we say the tertiary structure of a protein. And again, that's abbreviated as a 3 with a little prime up here. That is the 3D folded structure or folded form of the protein. And it's usually the functional form of the protein. And I wish I had a little cable here, but if I took a cable and I started to fold it up, here, I have one that I can kind of use, although it's attached to something. But if I took this little wire and I put it in straight chain, like this might be the amino acid or the protein in its primary structure. If I start to fold it up and wad it up and it becomes somewhat three-dimensional, and then I can do this, then I might have what's called the tertiary structure of the protein. Disregard this part, but I have a three-dimensional shape here, okay? And proteins will naturally fold up into a tertiary structure. Now, there's another structure called the quaternary structure. Oops, there's only one R in it. The quaternary, from cuatro, if you speak Spanish. And this is abbreviated as the 4 with a prime. This is when we have multiple tertiary proteins or tertiary structures bound together. Or sometimes we say multiple subunits. So let's say I have a protein that's, when it's folded up, makes a certain shape. If I take several of these proteins and I stick them together, then I get what's called the quaternary structure. There's multiple individual proteins that are bound together that form an even larger protein. Kind of like hemoglobin is made out of 2 alpha chains and 2 beta chains, and we can talk about that. So that would be the quaternary structure, is the 4th level of the protein folding, which would mean a bunch of individual tertiary structures, which are folded up, bind together into one larger protein. Okay? And some proteins are not active unless they're in the quaternary structure, like hemoglobin. Now, the process of a protein folding up is called protein folding. Protein folding is simply the process of a protein changing from primary to tertiary structure or form. It's the process of a protein folding up, going from the primary to the tertiary structure. Is a process called protein folding. Now I'm going to erase all of this, and I want to talk about one other thing. If I talk about a denatured protein, then that's the opposite of protein folding. So when I talk about a denaturing or a denatured protein, that's of a process changing from the tertiary to the primary structure. You can also think of it as unfolding of a protein. If a protein unfolds, like an enzyme, it can lose its enzyme activity or enzymatic activity. Okay? So those definitions are all important at some point, because later on in the semester when we talk about microbial metabolism and we talk about the breakdown of certain chemicals and food sources for certain bacteria, we can talk about how we treat the bacteria to slow their growth or control microbial growth and all sorts of other things. Now, this is why heating bacteria up and cooking them kills them, because it denatures all their proteins, amongst other things. Now, when a protein is folded up, so let's say that I've gone from a straight chain of amino acids and I have folded the protein and it is now in the tertiary structure, okay? And when I do that, I create these little pockets, and all along here are amino acids, and amino acids have chemical activity. They can bind to other things. Let's say I have some other molecule that's shaped like this. Not quite so, well, that one's too big. But let's say I have another molecule that's shaped like this, and these amino acids here, or these compounds, can bind there. So this is my enzyme. This would be called a substrate. A substrate is any substance acted upon by an enzyme. So before I finish this thought, let me explain something. In chemistry, if I have A plus B produces C plus D, this would be called my reactants. This would be called my products. In a chemical reaction. And if I need something to speed up or lower the activation energy, then that would be called a catalyst. Now, if this is a biochemical reaction, and this is an enzyme, then the reactants simply become called substrates. The substrate is whatever is acted upon by the enzyme. I still get products, so that's what a substrate is. So now, I have my enzyme and I have my substrate, and they can actually come together, and as I fold my protein up, it's not going to be folded exactly because I can't remember exactly how I drew it, and these two bind together so that the enzyme can act on it. I form what's called an enzyme substrate complex. And the enzyme substrate complex is simply the enzyme and the substrate bound together temporarily in the reaction process. So they form a complex. They come together. The enzyme is going to do something. For example, one of these amino acids might break some bonds in here. But before it does, I take my enzyme and my substrate. They form an enzyme substrate complex. And then at the end of the reaction, I'm going to get my enzyme out. Again, I can't remember exactly how I drew it. But now I might have two molecules here. I might break substance A, substrate A, into products C and D. So this would be A, this would be my enzyme substrate complex, and I might get two totally different compounds here. The area where the enzyme activity takes place, this little area is called the active site. So here's some definitions that you should know. You should know the definition of an enzyme. You should know the substrate is any substance acted upon by an enzyme. You should know that when they come together, they form an enzyme-substrate complex. That's the enzyme and the substrate bound together temporarily. And then as the chemical reaction continues, the enzyme, let's say this is a lytic enzyme or catalytic. A lytic enzyme would be something that breaks down. This is a catabolic reaction. I'm going to break this stuff down into two smaller subunits. So now, when the reaction's over, I get my enzyme back out. I get two products now because I broke one into two in a decomposition or in a catabolic reaction. And this would be considered to be the active site. And every time, if I had another one of these available, this enzyme could go back and do it again, and again, and again, and again, and just keep breaking it down until all of A is broken down into C and D. So the enzyme is not used up in the reaction and can continue to function. And if another molecule of A came along here and bound to the active site, it won't bind anywhere else. It binds to the active site. The active site is the region on the enzyme where the enzyme activity occurs, or where the substrate would bind. It's the area on an enzyme where the enzyme activity occurs, or where the substrate would bind. Okay? Also, if I were going to do a synthesis reaction, it might be a different enzyme, but let's say there's a different enzyme that could take these two and put them together and form this, and then the reaction could do that, then I would call that a synthesis reaction. Catabolic, anabolic, if it were a chemical reaction going in reverse. Okay? Not all chemical reactions are reversible. Okay? So let me make sure I've talked about all the definitions I want to talk about that are in my notes. One of the things that you should note, and I talked about this a minute ago, that the way that enzymes are named, so I hope you have all these notes. I'm going to erase all this, and I hope that makes sense in a very simplistic way of looking at it. Now, the name of the enzyme. How are enzymes named? They usually, not always, but they usually have ASE at the end of the name. Almost all enzymes are going to end in A's, like lipases, saccharidases, nucleases, proteases, peptidases. And usually, not always, but usually, the name can indicate the function. What is the enzyme function, or what is the enzymatic activity that takes place with this enzyme? I'm going to give you a series of enzymes that are examples of this. Excuse me. So, if I look at the following chemical reactions, and I have a whole list that are available for my students, and the rest of y'all could take some information from this, but there's a class of enzymes that fall under what are called oxidoreductases. Oxidoreductase performs what we call oxidation reduction reactions. And in chemistry, you sometimes hear these called redox reactions for reduction and oxidation. When we talk about reducing something, to reduce means make more negative. So in a reduction reaction, this is a reaction where the reactant receives electrons. So if I have some substance, let's say it's substance Q, and I run it through a chemical reaction, and I add some electrons, extra electrons to it, it becomes more negatively charged, so to speak, then I can call that a reduction reaction because I've made it more negative. Now, in that reaction, let's say I have substance Q plus substance P, and this has extra electrons. When I'm done, if I've given the electrons from P to E, this has been oxidized. And this has been reduced. So when I transfer electrons from one substance to another sometimes, and I'm not talking necessarily about ionic reactions, I'm talking about compounds that are just moving electrons around, then I can say this is a redox reaction. One substance has been reduced, the other substance has been oxidized. The oxidizer is the one that gives electrons. The reducer, the one that is reduced, is the one that has received the electrons. It's become more negative. This has become somewhat more positive. So oxidoreductases perform redox reactions. And I might have an enzyme here that transfers electrons from one to the other. Then this would be reduced, this would be oxidized. The chemical reaction would be called an oxidation reduction reaction. The enzyme that can perform it would be called oxidoreductase. And you can see from the name, it's given the function, does a redox reaction, it ends in ase, right? So now, I'm going to do another one, another type of enzyme, and I'm trying to make sure I go in the order that I wrote my notes. So if you see me look away, I'm looking at which one did I do next. A transferase. A transferase is an enzyme that will transfer a functional group. And if you've had chemistry, you should know what the functional groups are, like amino groups or carboxyl groups or hydroxyl groups, OHs and ketones and whatever, aldehydes. But it can transfer a functional group from one molecule to another. So if I have something that's called a methyl transferase, a methyl transferase is going to transfer, it's an enzyme that transfers a methyl group from one substance to another. So let's say I have substance X that has a CH3 on it. We call that a methyl group plus substance Y. If I put that in with a methyl transferase, and when I'm done, what I get out of it is substance X plus Y with the methyl group, then I know this enzyme was a methyl transferase because it transferred the methyl group from one molecule to another. So transferases can do that. They transfer a compound or a methyl group or some functional group from one molecule to another. Okay? Now, I'm running out of room, but again, you can see the function to transfer ase. Now, I'm going to erase all of this, but you're going to see this in the next few enzymes that I'm going to list. And later on, some of these enzymes may be important. I don't expect you to know them all right away, but over time, if you continue to see them, they'll stick to your brain. Another one that's important eventually to us is going to be called a hydrolase. Hydrolase will use water to lyse another molecule. Okay? It uses water to break up another molecule. So we call these hydrolases. Okay? And basically, this is called a hydrolysis reaction. Hydrolases perform hydrolysis reaction using hydro or water to lyse another molecule. And it's a type of decomposition or catabolic reaction. What's the next one I have listed for you? Lytic enzymes. There's a whole class of enzymes called lytic enzymes. And they perform lytic reactions, which means they break apart other molecules. So for example, this would be a lytic enzyme because it's going to lyse another molecule or break it down. And because it uses water, this would be called a hydrolytic enzyme or a hydrolase. Hydrolase is a hydrolytic enzyme or a lytic enzyme, and a specific class of lytic enzymes that use water. But all lytic enzymes can break down other molecules. Like lysosomes in eukaryotic cells are filled with lytic enzymes. Our white blood cells are rich with lysosomes. That's how they break down foreign invaders and kill infectious agents and things. A couple of others. There's one called an isomerase. An isomer means basically the same molecule, different shape. So an isomerase is simply an enzyme that can rearrange atoms on a molecule. It can rearrange atoms or functional groups on a molecule. Now, something you learn in chemistry is that dexter means right-handed, and I'm drawing a blank on the L. I can't think of what the L means, but it usually refers to a left-handed molecule. So I can have a molecule come in a D form or an L form. And if I have my hand with the fingers and all the same components, this would be the D form of the molecule. If I exposed it to a isomerase, the isomerase might flip the molecule around this way and move this functional group to this side. Then I've created an isomer. It's the exact same compound, but now I have this one functional group on the opposite side. I've gone from the D form to the L form, and we call those isomerases because an isomer of a molecule means it's the same molecule, but it's been rearranged a little bit. So we'll talk about D and L-type sugars and things in the future. So that's all that an isomerase is. It's an enzyme that rearranges atoms or functional groups on a molecule. It doesn't transfer them, it just rearranges them. Now, a couple of others. Well, the rest on the group I've already talked about. Saccharadases are enzymes that break down polysaccharides into monosaccharides. Lipases digest lipids into fatty acids and glycerol or into smaller compounds. Proteases and peptidases digest proteins, and nucleases digest nucleic acids. So we've discussed all of those before. Now, one of the things to know is that not all enzymes are proteins alone. Sometimes the enzyme requires other things to make it functional. Or, for example, one of the definitions I want you guys to know is called a proenzyme. This means before. A proenzyme is a non-functional form of an enzyme. And what I like to throw in there so that my brain can understand it is before it has been activated. So let's say I make an enzyme, fold up a protein, and I have an active site here. But sometimes part of the molecule is bound and covering the active site so nothing can bind there. So this is inactive. We would call this the proenzyme. And it's non-functional because it has not been processed yet. And sometimes the cell can process the enzyme, and what it will do is it will break our enzyme like this. And it'll cleave this little piece off. It'll literally cut a piece off and remove it, and now I get the active enzyme. And I call that enzyme activation, converting a proenzyme into a functional form of the enzyme. It's almost like pulling the pin on a grenade. If you were in the military and you carried grenades, you don't want the grenade to blow up on you. And so it's not active. But before you utilize it, or like this marker would be an inactive or a pro-enzyme. It won't do its job, which is right on the board. But if I cleave this part or cut this little piece off, and now my marker works. It is an activated enzyme. And so when we talk about enzyme activation, sometimes we talk about converting a pro-enzyme into an active form, into the enzyme. Okay? We talked a little bit about that in A and P. So the next definition I want to talk about is an apoenzyme. The apoenzyme is the functional portion of an enzyme. Now this is not going to make a whole lot of sense until I define some of the other terms, but if I have a proenzyme, once I cleave it, the functional part would be called the apoenzyme. Okay? Sometimes enzymes require other factors or other molecules to activate them. One of them is sometimes called a cofactor. And the best way to define the cofactor is that the cofactor is usually some kind of inorganic compound, like an ion, that increases the enzyme function. So sometimes I have an enzyme that's all folded up, but the active group is not necessarily, or the active site is not necessarily exposed or in its final form. Now, I think of enzymes this way. If I asked you to carry 2 basketballs under your arms, and I said, if you carry these basketballs for the next 12 hours and you do not drop one, you get 100 on every test for the rest of your career and automatic job making $150,000 a year. And if somebody walked around and threw things at you, like a marker or an eraser, you're like, You're not going to let go of these 2 basketballs because you want your grade. But what if somebody threw you a live baby? What would you do? Well, it depends on how badly you need. No, I'm just kidding. What you would do is you might let go of these substances and grab that baby. And that's sometimes what an ion can do, or an inorganic molecule can come in and something can bind to one side of the molecule here, and that'll cause these chemical bonds inside to change and the protein to change its shape. And so when the protein has this substance bound, it might change its shape and get its active site. So we call that a cofactor. It's usually an inorganic molecule. It's not necessarily a compound, but an inorganic molecule, sometimes an ion, that can increase the enzyme's activity. And basically what it does is it can change the shape of the enzyme in another area that opens up the active site. A similar molecule is called a coenzyme. And one of the only differences between a cofactor and a coenzyme is that coenzymes are usually organic molecules that increase the enzyme function. So we have some molecules that have carbon in them that can play a major role in this. And these can be things like NAD and FAD and NADH and FADH2 and FADH, nicotine, adenine, dinucleotide, some organic compounds. So the only difference is these are inorganic, we call it a cofactor. If it's an organic molecule, something that has carbon in it, then we call it a coenzyme. And those coenzymes can increase the enzyme function. Very similarly, I might add. And finally, the last thing I want to talk about would be the holoenzyme. And when I have an enzyme, and I have whatever that enzyme needs, its cofactor or its coenzyme with it, and it's functioning, we call that the holoenzyme. So it's the entire enzyme with its cofactor or with its coenzyme in its functioning form. Okay? Now, I'm going to erase all this. A couple of other concepts I got to get to, and hopefully we can wrap this lecture up and we can move on to energy metabolism in another lecture. I'm going to break these up because I don't want them to get too long. We could talk about metabolism for weeks and weeks and weeks. So, one of the things that can affect enzymes, so things that can affect enzyme activity. And I'm not going to talk about everything, but I'm going to talk about a few things. One of them is temperature. Too high of a temp can decrease enzyme activity. It can denature the enzyme. Sometimes it can kill the enzyme activity. If I have too low of a temperature, this slows the rate of chemical reactions. This is why we put things in the refrigerator. It slows the rate of the chemical reactions that would break the food down and make it spoil, so to speak. So, essentially, all enzymes have an optimal temperature at which they work, at which they function. And if I raise the temperature too high, then I can kill the enzyme activity by denaturing the protein. If I lower the temperature too low, then all the other parts of the chemistry slow down. Chemistry happens more slowly because we're talking about what's called Brownian motion. The molecules are vibrating around and moving, and if they vibrate more slowly and move more slowly, then it takes longer for them to come together and form a chemical bond or break a bond. So temperature can affect enzyme reactions. There's an optimal temperature in a small range. Like in our body, a lot of the chemical reactions are happening at 37 degrees Celsius or 98.6 degrees Fahrenheit. If we get too hot or too cold, it will change the rate of the chemical reactions. It can slow them or stop them, and you can die. Now, the same concept applies to pH, which is how many hydrogen ions are available here, or the likelihood that there's some hydrogen ions would be available. Even if I added them, if I had something that is too basic, it'll suck up those free hydrogen ions. So if I have too high of a pH, meaning too basic or alkaline, or I have too low of a pH, too acidic, it can slow the rate of the chemical reaction or kill the enzyme activity. Okay? And in many instances, both could denature the enzyme. Because after all, when two different amino acids come together in a chemical bond, a lot of times this one has an extra hydrogen and this one has an OH, an alcohol group. And they can come together and release water. If I put a whole bunch of little hydrogen ions, if I put this in a very acidic environment, what's the likelihood that one of these hydrogen ions might bump in here, bind to this amino acid, and break this bond and denature it and reverse the reaction? And conversely, what if I put, that would be too acidic. If I'm too basic, then I'm going to have a whole lot of OH negatives floating around. And one of those could bind to this amino acid and denature the protein. So all enzymes have an optimal pH at which they function. If the pH is too high and too basic or alkaline, it can kill the enzyme activity. If it's too low of a pH or too acidic, it can kill the enzyme activity by denaturing the enzyme. Okay? And another thing that can affect enzyme reactions is what we call substrate concentration. There's an optimal concentration at which the enzyme will be working efficiently and producing a whole lot. So let's say I have some chemical reaction I want to perform here. And I need to turn substance A and substance B into C, 2 amino acids into a dipeptide or something. Well, what if I don't have any of B around? What if I have a whole lot of A but very little B? If I've got 100 of these and only 2 of these, how much product am I going to get? So the concentration of the substrates can affect the rate of the enzyme reactions. If I don't have enough substrate, The enzyme reactions won't happen rapidly. There won't be enough of them. I won't get enough product. Now, what if I have 10 million of these and 10 million of these? Extremely high concentrations of them. Well, at some point, my enzyme might bind substance A and substance B and release them as some larger molecule, substance C, like this. Okay, with a little chemical bond here. But at some point, if I have so much of this, I can overwhelm the enzyme. It becomes so saturated. Saturation of the enzyme means I've bound them all up. The enzyme reaction can't happen any faster. So as I increase the substrate concentration, I can increase the rate of me producing a product up to a maximum. Eventually there becomes so much substrate that I saturate the enzyme, and it's like you have two hands. And if I had a conveyor going by here and I asked you to put cakes in a box, there's only so fast that you can work. And if it's going too fast, you can only get so many done, but some of the cakes are going to fall on the floor, not in a box. And so we call that saturation, overwhelming the enzyme with too much of the substrates. Okay? And finally, the last concept is called enzyme inhibition, or it's called negative inhibition, and some people call it, you can stick the word negative feedback inhibition. So essentially what happens here is sometimes I can take some substances and run them through an enzyme reaction to get product C. But as product C starts to build up, it can feed back and bind up the enzyme and inhibit the enzyme from functioning and shut itself off. And so we call that negative inhibition or negative feedback inhibition, where the product feeds back and shuts down the enzyme reaction. Or in some instances, it could come up and bind up the reactants, the substrates, and then the reaction will shut down. Okay? Now, there's two ways that I can inhibit the enzyme. So let's say I have an enzyme like this, and here's its shape, and here's its functional, its active site here. Now, if I can take substance A and substance B and bind them here and turn those into a much larger substance C, what if I introduce another compound, some other stuff, some other molecule that can fit in this active site? So that when this thing fits, then substance A cannot bind. I'm blocking A from binding to the active site. Then I'm competing for the active site. So we call that competitive inhibition. To inhibit means to stop it from functioning, and in competitive inhibition, something competes with the substrate for the active site. So if I have some competitive inhibitor that I can introduce and I can stop a chemical reaction in an infectious bacterium, then I can stop the bacterium from making some toxin or something that makes me sick. Then I can use competitive inhibition. I can introduce some substance that binds to the active site and blocks it or competes with that substrate for the active site and stops the enzyme reaction. Another form of inhibition is called non-competitive inhibition. In non-competitive inhibition, the inhibitor binds to an alternate site from the active site. Essentially, it doesn't compete. It doesn't inhibit the enzyme by binding to the active site. What if I have some substance bind right in here and force these amino acids in this part of my enzyme to bind to it. And when that does that, what it does is it might change the shape of the active site so that only substance B can bind, but now substance A can't bind. As long as this thing is bound in here, it's preventing that substrate from interacting, but it's not binding to the active site itself. We call that non-competitive inhibition. It's not going to compete with the substrate for the active site. It binds to some alternate area that can alter the active site, but doesn't bind directly to the active site. During competitive inhibition, whatever the inhibitor is, it will compete with or bind to the active site and block the substrates from binding. I hope that makes sense to you. Now, when we're talking about all of this, one of the reasons we're talking about some of these things is because we have to understand how we can affect microbial metabolism. Ultimately, microbes need energy to perform a number of activities. So, whether it's prokaryotic cells or eukaryotic cells, cellular activities that require ATP. Well, protein synthesis is one of them. In order to synthesize proteins, the ribosome and the tRNAs will also use energy, ATP. If I run out of ATP or I can inhibit energy production in a bacterium, I can stop protein synthesis. And I wrote protein synthesize, but that. Cell division. Mitosis, if it were a eukaryotic cell, binary fission in bacteria. If I can inhibit energy production, I can stop cell division, I can decrease the bacterial growth rate so that maybe the infection can be killed by my white blood cells. Active transport. If you watch the videos to review osmosis and diffusion and active transport and tonicity, one of the things that we know is that when I have a bacterium, some bacteria have a very thick cell wall outside of them made out of proteoglycan. And one thing that cell wall does is it prevents lysis of the bacterium. Because if I took a cell that does not have a cell wall and I put a whole bunch of salt inside the cell and I made the inside hypertonic and the outside only has a little bit of salt and it's hypotonic out here, then water is going to move. Since the proteins can't cross, the water will move into the cell, it will swell, and eventually the cell will rupture and kill the cell. Well, one of the reasons that bacteria can survive a hypotonic condition is that even though they're hypertonic inside with all these proteins and ribosomes and other substances, the cell wall prevents them from rupturing. So even if water moves in, the cell wall will prevent them from popping. And some chemicals, like lysozyme in our tears, can actually poke holes in the cell wall. And as they take on water, bacteria can lyse. And so lysozyme ends up having a lytic effect on bacteria by breaking down the cell wall, causing them to lyse due to the fact that they are hypertonic to the surrounding environment. They will take on a lot of fluid and rupture. Okay? So, now that's all passive transport. But in active transport, one of the things that we know is that these bacteria sometimes have to get sugar into the cell. And sometimes they have to get waste out of the cell. And some of these substances are moved by active transport. So moving things into and out of a cell requires energy. So we have protein synthesis, we have cell division like mitosis and binary fission. DNA replication is part of that. So in order to divide, split in half, we also have to replicate the DNA. Also part of protein synthesis that requires ATP is the conversion of the DNA into messenger RNA, that process of rewriting DNA into mRNA, moving things around inside the cell. So there's a number of things inside the cell that require energy. Now, the reason all of this is important is because in the next lecture, what we're going to talk about is we're going to talk about bacterial metabolism. We're going to talk about eukaryotic cell metabolism and then compare that to prokaryotic cellular metabolism. And we're going to look at how can we disrupt metabolism, because this is how a lot of antibiotics work, is some of them disrupt energy production or enzyme function. This is why heating up bacteria or cooking your food can kill some of the bacteria in it, or putting things in the refrigerator and slowing down the chemical reactions can slow bacterial growth. And as long as the bacteria are too few in number, it won't spoil the food. But if you leave it out, the bacteria grow rapidly at a higher temperature, and at room temperature, so to speak, and they can cause things to rot or spoil much more rapidly. And so our knowledge of microbial metabolism and energy production and bacteria and their enzymes, which enzymes they have in order for them to produce energy, gives us an avenue for treating the bacteria in a way, and other prokaryotic cells in a way that are beneficial to mankind, prevent food spoilage, or take advantage of some things that they do, for example, for production of ethanol and biofuels and beer and wine and other things. So we're going to talk about that in another video. I hope you guys have a basic understanding of the 4 major organic compounds and how they function, what we use them for in cells, both prokaryotic and eukaryotic cells. You need to know the primary function of each of the 4 and know that all 4 can be converted into sugar for energy. You need to understand what proteins and what enzymes are, what the active site is, what cofactors and coenzymes are, and all of this terminology. And hopefully this little lecture makes it make a little bit more sense rather than just reading an encyclopedic list of structures and definitions. Look, I hope that you're starting to understand some of this. Right now we're building up to start utilizing all the information that we've talked about in all these lectures to really understand the world of microbiology. I hope you had as much fun as I did. I hope you learned something, and I hope to see you in the next video. And if you're watching these on YouTube, please hit like and subscribe, or at least hit like and let me know if you're liking these videos. Talk to you in the future.